So now we have a question. What would happen if we increased the dose of an agonist? What would happen to the response or the action that this agonist do? What if, for example, this agonist increases the heart rate like adrenaline? If we gave a certain dose, let's say it's dose X, and it increased the heart rate by 30 to 40 beats per minute. If we doubled this dose and we made it 2x, we would expect that adrenaline would increase the heart rate by double of this 30 to 40 beats per minute, and it would increase it by 60 or more beats per minute. So we would expect that by increasing the dose, we would have an increase in response. And we can draw that relationship on a curve or a graph by plotting log dose, not the dose for mathematical reasons, versus the response. And we would get this sigmoid curve. As we can see, by increasing the dose, we found that there is an increase in response. And this response is increased until it reaches a plateau, a point where there is no increase in response. There is a maximum response. And we call that response the E max, or the maximum efficacy of a drug. This is an agonist. This is a curve for an agonist because there is an activity, there is an intrinsic activity that increases by increasing the dose. But what about this curve? This curve is for a ligand that also has a response or has intrinsic activity, but it's somehow lower than the intrinsic activity of the agonist. What is this curve for? Yes, it's for partial agonist or partial agonist antagonist. And we have here a third ligand that has no response. That is, yes, the antagonist. Okay. And we have a fourth type of ligand that we wouldn't talk much about it. It gives a response that is lower than the normal, or it decreases the normal response of a certain receptor when binding to it. And for that we call that ligand inverse agonist, because this ligand does exactly the opposite of an agonist. Okay, so if we have a certain receptor that increases heart rate, when this ligand or inverse agonist, when bound to the receptor, it causes a decrease in heart rate. Okay, we wouldn't talk too much about this type of ligands. Just we would like to know that there is an a ligand that is called an inverse agonist. Okay, So that was a drug or dose response curve. A dose response curve. That was a curve between the dose and the response. So now let's draw that again to discuss some important points. Again, we draw a curve between log dose and response. Okay, and here we have a drug. This drug is an agonist. There is a response. Okay, this drug A, here we can find its Emax or the maximum response. Okay, what if we extrapolate this point here to here? First, what is this point? This point here is half the maximum response. This is half the Emax. So if we extrapolate this point to the dose, we get the dose that give us half the Emax or half the maximum response, and we call that dose EC50. EC50 is the dose that give us half the maximum response. Okay, now let's have a look at this curve here. This curve here is for an agonist also. We call that drug, let's call it drug B. As we can see, this drug B gives the same Emax and the same half the Emax of drug A, but drug B give this half Emax with higher EC50. We needed larger dose from drug B to get the same response we get from drug A. Okay, drug B has larger EC50 than drug B. So we call drug A, or say that drug A is more potent than drug B. So we compare the potency of drugs by comparing their EC50. EC50 of drug B is more than the EC50 or larger than EC50 of drug A. So we could get the same response by lower dose. That what would make a drug more potent. Okay, and here we have drug C, but drug C has lower efficacy than drug B and A. So drug A and the drug B have the same or could reach the same Emax, but drug B could reach it after giving larger dose than drug A. So drug A is more potent than drug B, but drug C couldn't reach that maximum efficacy as of that of drug A and drug B. Okay, now we have a question. This Emax or the maximum response, when could we reach it? Okay, that Emax or maximum efficacy is reached when all the receptors of the cell is occupied by the agonist. So when all the receptors are occupied, there is no more response when an agonist is given because there is no receptor that this agonist could bind to. So we reach this Emax when all receptors on the cell are occupied by the agonist. So in order to get the maximum efficacy or the Emax, all, all the receptors of the cell are occupied by the agonist. But there is an exception to this rule. We have some cells that reaches the maximum response or the Emax without its receptor being totally occupied. These cells reach the maximum response without full occupation of the receptor. They still have receptors that are not occupied, and these receptors are called spare receptors. Okay, so spare receptors are receptors that are on the cells that is not occupied by the agonist, and despite that there is a maximum response in the cell. Okay, now we have to discuss the last point in our introduction of pharmacodynamics, which is when we give a drug that is an agonist on a certain receptor, our body tries to resist the action of this drug, and that happens through decreasing the synthesis of this receptor. So the our body tries to keep what is called homeostasis. It tends to um, make the environment constant by decreasing the synthesis of that certain receptor. And this is called down regulation. And on the other side, if we give an antagonist, a blocker for the receptor, our body increase the synthesis of this receptor, and this is called upregulation. So we have upregulation in which our body increases the synthesis of a certain receptor when we give antagonist for long time, and we have downregulation in which our body decreases the synthesis of a certain receptor when we give an agonist for large time.